Thank you. Okay, the next session, we're going to focus on Barrett's esophagus for the next 2 talks and case presentations. So let us begin with some of the molecular mechanisms of carcinogenesis in Barrett's esophagus. So as you know, our current management of cancer risk in Barrett's esophagus is to perform surveillance for dysplasia. But dysplasia is an imperfect predictor of cancer risks for a number of reasons. Dysplasia is a constellation of histologic abnormalities, which suggest that clones of cells have DNA damage, rendering them neoplastic and predisposed to malignancy. Therefore, dysplasia is the histologic expression of DNA damage that precedes malignancy. The molecular changes that allow a cell to progress to dysplasia and cancer occur before the histologic detection of dysplasia. So for example, here are 2 metaplastic cells, and histologically, they both look the same. However, this cell has sustained DNA damage, giving it a growth advantage. As the cell acquires more DNA damage, and it becomes dysplastic, and eventually with even more DNA damage, the cell becomes cancerous. So while there are no clinical or histologic characteristics that can distinguish this cell from this other metaplastic cell, there are molecular characteristics that could tell you. So as Barrett cells undergo DNA damage that allows them to progress to cancer, they acquire core physiologic features of cancer. So to become cancers, Barrett cells must acquire the ability to provide their own growth signals, to avoid growth inhibitory signals, to avoid apoptosis, to replicate without limit, to sustain angiogenesis, and to invade and metastasize. So let's first look at how Barrett cells might be able to provide their own growth signals. So in general, this occurs through activation of oncogenes. Proto-oncogenes are normal genes that stimulate cell growth. Oncogenes are just proto-oncogenes that have now become overactive as a result of mutation. And examples of oncogenes and oncogenes lead to uncontrolled cell growth, and examples of oncogenes that have been implicated in Barrett's carcinogenesis include cyclins, such as D1 and E, and growth factors in their receptors, such as transforming growth factor alpha, epidermal growth factor, and the epidermal growth factor receptor. The next core feature that Barrett cells must acquire as they progress to cancer is the ability to avoid growth inhibitory signals. And in general, growth inhibitory signals are transmitted by tumor suppressor genes. And these are normal genes that restrain cells' ability to grow. Tumor suppressor gene inactivation enables cells to avoid growth inhibitory signals. And examples of tumor suppressor genes that are inactivated as Barrett's progresses to cancer include P53, P16, and the adenomatous polyposis coli gene. And there are at least 3 ways to inactivate tumor suppressor genes. This includes loss of heterozygosity. Usually that accompanies mutation in the remaining allele. And there's also gene promoter hypermethylation. So let me show you how these 3 different mechanisms work to inactivate tumor suppressor genes. Let's first look at loss of heterozygosity. So here's a chromosome with 2 alleles. One allele is from the mother and the other is from the father. And on each allele, there's a copy of the tumor suppressor gene. with loss of heterozygosity, one allele is missing, leaving only one remaining copy of the tumor suppressor gene. If this copy then becomes mutated, then the gene is inactivated and you lose function of a tumor suppressor gene. Another common mechanism is that of gene promoter hypermethylation. So here's a stretch of DNA, and upstream of that arrow is the gene promoter. The arrow indicates where the DNA is made into messenger RNA. Upon binding by a transcription factor to the gene promoter, and in this morning's lecture, when I talked about NF-kappa B and AP1, those were transcription factor binding sites in the DNA. It stimulates the production of mRNA in the genus produced. If you have gene promoter hypermethylation, there are methyl groups that are sitting in the promoter of the gene, such that the transcription factor is blocked from binding, and then no mRNA is produced, and the gene is now inactivated. Another core feature that Barrett cells must acquire as they become cancer is the ability to avoid apoptosis. Apoptosis is a genetically regulated form of cell suicide. And in general, apoptosis is good because it's going to eliminate cells that have extensive DNA damage, therefore the cell can't replicate and cause sustained or lasting mutations. There are a number of ways in which to trigger apoptosis. For example, it can be activated by DNA damage or mutation, and this is usually through p53. The apoptosis pathways could also be activated through death receptor activation with their ligand. And in Barrett's, we see this with FAS and FAS ligand. There's also metabolic abnormalities that can inhibit these pathways to apoptosis. And again, the example in Barrett's is overexpression of COX-2. COX-2 can act to inhibit these commitment to die pathways. Regardless of how the pathways are activated, they converge on a single executioner pathway, which triggers cell death. Now let's look at another hallmark feature, the ability of cells to replicate without limit. So normally, as cells undergo successive divisions, they reach senescence. And senescence is an intrinsic mechanism of cells that limits their proliferative capacity. And this involves the loss of telomeres. Telomeres, as shown in this cartoon, are these disc-like structures that are located at the ends of chromosomes and that are lost with each cell division. When these telomeres become too short, it triggers the cell to undergo senescence. Telomerase is an enzyme that synthesizes and then stabilizes telomeres, and stable telomeres can confer immortality to the cell. So this morning's lecture, when I was showing you those esophageal squamous cell lines that we made in the lab, this is the enzyme that we put in these normal cells in order to avoid our cells from undergoing senescence so they could continually proliferate and become immortal in the culture dish in the lab. If I had a cancer cell from a patient that I wanted to put in the lab, I wouldn't have to go through this process because the cancer cell does this on its own. Another hallmark for Barrett cells to become cancer is the ability to sustain angiogenesis, which is the synthesis of new blood vessels. So this cartoon illustrates how cancer cells go about sustaining angiogenesis. Tumor cells themselves can secrete vascular endothelial growth factor, as well as other growth factors to support angiogenesis. But vascular endothelial growth factor is the most well studied. This growth factor can then bind to its receptors, the vascular endothelial growth factor receptors that are located on the endothelial cells, and signaling by VEGF stimulates the growth of new blood vessels so that they can facilitate further growth and amplification of tumor size. The final core feature is the ability of these cells then to invade and metastasize. And although the exact mechanisms of invasion and metastasis are poorly understood, they do involve changes in cell adhesion molecules, such as catenins and cadherins. So if we look closer here at this cartoon of the normal tissue, and you slice it, cut it, and open it up, and look here at the junctions between the cells, you can see that cadherins form these cell-to-cell bridges, protein bridges between the cell. And they're anchored to the cell's cytoskeleton through catenins. Alterations in either catenins or cadherins, as you can see, would allow the cells to dissociate from each other, and they would lose their cell structure. Invasion involves now the secretion of matrix metalloproteases. These allow the cell to degrade extracellular matrix. And metastasis then involves the cell changing its phenotype, which is also termed epithelial mesenchyme transition. And this allows the epithelial cells to take on features of stromal and mesenchymal cells, and one of those key features is now the ability to migrate. So not only can the cancer cells degrade the matrix, but now they have the ability to now creep through, get into the circulation, and metastasize. So when cells appear histologically in tissue biopsies as metaplasia, some of these cells, in fact, have acquired an increase in cyclin D1, transforming growth factor alpha and epidermal growth factor receptor. There's loss of P16. There's loss of P53, and there's an increased expression of COX-2. They do express VEGF and VEGF receptors, and they have the ability to synthesize and secrete matrix metalloproteases. When we have a cell that the pathologist says to us is dysplasia, in fact, those cells have an additional increase in cyclin E expression. There's loss of APC. Telomerase is often found to be activated in high-grade dysplasias. There's a decrease in E-cadherin and catenin staining at the surface of the cell. The bridges are destroyed. When cancer is found, again, there's a further increase. There's KRAS mutations, epidermal growth factor overexpression. The cells express fast ligand, so they can, in fact, kill the tumor-infiltrating immune cells. So they're providing protection against these cells that are there to destroy them. So as you can see, just by simply looking at this very watered-down genetic model, that some of these metaplastic cells, in fact, have acquired a number of molecular abnormalities that give them growth advantages of neoplastic cells, even before they exhibit any histologic features of dysplasia. So in the last few years, there's been more progress in sort of understanding the complexities of cancer formation, and there are two new emerging characteristics of cancer cells. These are considered emerging because while they do appear to be important for cancer development, it's not entirely clear that the cell has acquired these new emerging characteristics independently, or rather that they're simply just a result of already having acquired the 6 core features that we just discussed. Nevertheless, these emerging characteristics include the ability of cancer cells to avoid immune destruction and the ability to reprogram their energy metabolism in order to support the continuous proliferation and growth of the tumor cells. So I just wanted to take a minute to look over and sort of go through this reprogramming of cellular energy metabolism. And in the literature, you often see it referred to as the Warburg effect. So normal cells process glucose to pyruvate in the cytosol via glycolysis, and then it goes into the mitochondria where CO2 is produced. This is a highly energy efficient process. In contrast, Warburg noted that cancer cells seem to reprogram their energy metabolism by limiting it largely to glycolysis. Glycolysis is less energy efficient, and it requires an enormous amount of glucose uptake by the cell. And this characteristic of the cancer cells is what we used when we're looking at PET scans. And PET scans use a radio-labeled analog of glucose, this FDG, to detect the cancer cells, because the cancer cells have this huge dependence on glucose uptake. So the scan is positive in the cancer cells, so you can distinguish it from the normal cells, which don't have such a heavy dependence on glucose uptake. And in fact, FTG has been used in some early studies for in vivo molecular imaging of Barrett's, dysplasia, and cancer, again, based on this Warburg principle. There are also two enabling characteristics of cancer cells that help them acquire those 6 core features that we discussed. And this includes tumor-promoting inflammation, which in fact seems to inadvertently assist the cell in acquiring these other features. And there's also genomic instability and mutability, which is thought to drive this whole process of tumorigenesis. And in fact, the finding of genomic instability and mutability has been shown to be one of the most promising biomarkers for use in Barrett's esophagus. So one marker of genomic instability, or a way to detect it, is through aneuploidy. And another biomarker that has shown promise for clinical use is 17p LOH. And 17p is the chromosomal site where the p53 gene is located. So let's first look a little bit more about how aneuploidy. So aneuploidy reflects the accumulation of multiple genomic abnormalities. It does not indicate a single genomic abnormality like the ones that we just discussed. Aneuploidy refers to an alteration in the cellular DNA content from the normal 2n or 4n, where n is the number of chromosomes in the cell. And I'll show you a little bit illustration on what exactly I'm talking about. Aneuploidy indicates that the DNA is unstable, and it's at an increased risk of neoplastic progression. Aneuploidy can be detected by FISH, by flow cytometry, and by automated image cytometry. So here's an example of aneuploidy that's being detected by FISH. Here you can see they're using a yellow probe for chromosome, actually green probe for chromosome 9. And there's a yellow probe that's going to look at the specific chromosome area that P16 is located on. And you can see in this cell, there's 3 copies, or 3 green fluorescents and 3 yellow. Normally, there should be either 2 copies or 4 copies. But since this is 3, this reflects aneuploidy. Another way to detect aneuploidy is by flow cytometry and or automated image cytometry. And both of these techniques are going to detect the DNA content within the cell. So in this flow cytometry tracing, this is from a biopsy of a normal tissue. The y-axis is going to look at the number of cells that are contained in the tissue, and the x-axis is going to look at each of those cells and then tell you what the DNA content is in each of the cells. So in this normal tissue, you can see there's a lot of cells that have a 2N amount of DNA, and there's a smaller proportion of cells that have a 4N amount of DNA. And this is to be expected in a normal tissue. So on the left, again, is this normal flow cytometry tracing. And then on the right is now a flow cytometry tracing from a patient with Barrett's esophagus and high-grade dysplasia. Took the biopsy, run the cells in the flow cytometer, number of cells on the x-axis, and the amount of DNA is on the y-axis. You can see here that the 2N and 4N peaks are quite small compared to the normal tissue. And there are these 5 additional peaks detected by the flow cytometer, which tell you that there's a large amount of cells that have other than the normal 2N or 4N amount of DNA. These peaks represent the aneuploid populations, and it indicates that the tissue is at an increased risk of neoplastic progression. Now, here are data using flow cytometry. And I know these data are old. They're by Brian Reed at Fred Hutch Cancer Center. They've been around a long time. And the AGA recently put out a medical position statement, and I reviewed sort of all the molecular events that are coming out. These are still the best data out there on how these molecular markers have the ability to perform. So I thought I would just show you the classic studies. In this example, he looked at aneuploidy, and he also looked at an increase in 4N. And an increase in 4N is also abnormal. There's a certain amount of 4N a tissue should have, but if you have over that, it's also considered a marker of genomic instability. They looked at these biomarkers in patients whose biopsies had low-grade dysplasia or just showed no dysplasia, so non-dysplastic Barrett's metaplasia. They took biopsies during surveillance. They then ran the markers on the first initial biopsy, and they followed patients over time. And they had biopsies during the progression to cancer. And what they found is that patients without aneuploidy and a normal 4N had a very low rate of cancer progression at 10 years. However, if the biopsy showed either aneuploidy or an increased 4N, the rate of progression to cancer at 10 years approached 50%. So in addition to aneuploidy, the other marker that has shown the most promise for clinical use is this 17p LOH. Here are data, again, coming out of Brian Reed's group in Seattle, looking at 17p LOH as a biomarker to predict progression to cancer in Barrett's patients. In this study, patients were enrolled in a surveillance protocol again. Biopsies were done at the first session. Patients were followed over time to see who did or did not develop cancer. And they looked at LOH. In patients without LOH, again, there was a low rate of progression to cancer. However, if the biopsy showed LOH, the rate of progression to cancer at 5 years approached 60%. And then here are recent data from a case-controlled study that looked at P53. This time they were detecting P53 by immunohistochemistry, and they combined it with the finding of low-grade dysplasia on histology. If you look at low-grade dysplasia, the positive predictive value in predicting esophageal cancer progression was 15%. However, if they combine that with aberrant P53 expression, which meant P53 was either overexpressed, as shown here, or there was loss of P53 expression, either one of these, combined with low-grade dysplasia, had a positive predictive value of 33% in detecting those that were going on to esophageal adenocarcinoma. And I really wanted to show this to you because the British Society of Gastroenterology is coming out with their new guidelines. And when you read their guidelines, they're actually going to suggest considering adding p53 immunostaining to your histologic diagnosis in order to improve the ability to stratify those with dysplasia on who's going to progress to cancer and who might not. And then finally, here's some data now combining these biomarkers that we just talked about. They combined 17p-LOH, DNA content, which is aneuploidy, or 9p-LOH, and again, 9p-LOH is where the p16 gene is located. You can see here that as the number of genetic abnormalities in the tissue sample increases, so does the risk of cancer progression. At 6 years, patients who demonstrated all 3 genetic abnormalities had an 80% incidence of cancer, compared with 0% in those without any genetic changes. And in the studies I've just shown you, some of the cells looked normal to the pathologist. Remember, in the first aneuploid study, those were patients that had non-dysplastic Barrett's metaplasia. And yet they had aneuploidy, they had an increased 4N, some of them had LOH. So in fact, it does appear that the biomarkers seem to be much better than the histology in predicting cancer progression. But it's clear from here that they're still far from perfect. But what if we did have a perfect biomarker? One that was 100% sensitive and 100% specific, which is what we're all trying to strive for, is this perfect biomarker. How effective would that biomarker actually be? Well, here's a Markov model that John Inadomi and Joel Rubinstein did in Washington, actually looking at the question. So what they did is they modeled a cohort of 10,000 50-year-old white men with GERD symptoms, and they followed them for 30 years. During that 30 years, this model predicted that if we did no surveillance, 366 cancers developed, and all but 10 of them died of their cancer. If surveillance is done to detect dysplasia, which is what we do now, only 315 cancers developed, which was a decrease of 14%. And since the cancers were detected early, only 157 of them died of their cancer. Instead, what if we used our perfect biomarker, one that was 100% sensitive and 100% specific? You would think that we might be able to eliminate all the cancers. But in fact, this Markov model predicted that it only decreased cancer development by 53% and cancer deaths by 87%, not 100%. So even a perfect biomarker is better than dysplasia in predicting cancer deaths and in decreasing cancer deaths. But it's important to appreciate that even a perfect biomarker is not going to eliminate cancer deaths. So I just showed you individual biomarker performance, and then I showed you a panel where we had 3 biomarkers in one. But recent technology now has allowed us to assay hundreds and thousands of biomarkers all in one panel. So more of the studies coming out now are going to be looking at these arrays. And the ones that have been shown in Barrett's to have some utility are gene expression or microRNA arrays, mRNA arrays. There's microRNA expression arrays, protein expression arrays, and methylation panels. So let's first look here at this mRNA expression array, and they're all going to function the same way. The genes on the array may differ from study to study, but the technique and what they're looking for is not going to change from study to study. So in this example, this array has 18,000 genes per sample, and the expression of 18,000 genes per sample was done on cancers and on non-dysplastic Barrett's esophagus. An increase in gene expression is shown in red, and a decrease in gene expression is shown in green. And you can just see you don't even need to know the names of the genes on the panel, but just looking at the expression, you can see there's differences in red and green expression between the cancers and the non-dysplastic Barrettts. And just looking at the color pattern, the investigators were able to separate esophageal cancer from that of non-dysplastic Barrettts. In a lecture presentation presented this year at DDW, Rebecca Fitzgerald's group presented these data using a microRNA gene expression. Here she just assayed 90 genes. So you can see that this technology is now allowing you to considerably narrow down the types of genes that you can assay for and still separate dysplastic here, non-dysplastic from dysplastic Barrett's esophagus. And rather than using expression of mRNAs, this is the new kid on the block, and I think most studies are going to be talking about using microRNA expression arrays. So I just wanted to give you some information about what they're actually talking about when you're looking at microRNA. These are non-coding RNA molecules, and they're about 18 to 25 nucleotides, so they're really small RNA molecules. These microRNAs can inhibit translation or promote degradation of messenger RNAs that have complementary sequences to them. Each microRNA can interact with many messenger RNAs, and each messenger RNA can be regulated by multiple microRNAs. So there's no one-to-one. These microRNAs seem to be pretty promiscuous, and there's lots of target mRNAs. MicroRNAs can target oncogenic and or tumor suppressor mRNAs. So some of these microRNAs can help form a tumor and others can help prevent a tumor. And microRNA targets can influence proliferation, apoptosis, invasion of metastasis, which are some of those core physiologic features we talked about earlier. So here's another type of array, and this is an array of a gene methylation panel. So this technique is going to look at the amount of methylation on the promoters of various types of genes. Remember, hypermethylation is a way to turn off the mRNA expression. So in this study, they used a repository of clinical samples from Barrett's patients that progressed to high-grade dysplasia and cancer, and from Barrett's patients that didn't progress, so progressors and non-progressors. They used a hazard ratio index, and this index included clinical characteristics such as age and then the length of the Barrett segment. And then they did a methylation panel, and they looked at the methylation of 6 genes. And they call this their hazard ratio index, and this was their biomarker. What they found is that in patients who progressed to high-grade dysplasia or cancer, which was this group, that this hazard ratio index exceeded a value of 5 within 2 years before the patient clinically manifested with their high-grade dysplasia or cancer. So if you're going to develop a biomarker, you want one that's going to turn positive before your patient has the disease. And so this had pretty good performance as a predictive biomarker panel. Recently, this came out of the lab of Steve Meltzer, who I trained with at University of Maryland. I recently got an email. A methylation panel similar to this is now commercially available. If you do Barrett's esophagus and like methylation, it's out there. And I can't remember the name of the company that recently contacted. There's the company name, but you can actually order this. I don't know what you're going to do if you order it and get the result. There's no guidance on what we're supposed to do with it, but you can actually order this now. So for all the biomarkers that I just shown you, tissue biopsies are obtained, you take the biopsy out, you throw it in a dish, you put it in a test tube, and you assay the biomarker. What I want to show you is sort of where we're headed, I think, in the future, is the ability to do this in vivo. And usually when you're doing this in vivo, you're going to give off a fluorescent signal, and lots of times you're going to be picking it up with fluorescent tags, which are often green. So in order to do this, you're going to need an endoscope that can collect fluorescence images, And at this point, you either need a fluorescence endoscope or a confocal laser endomicroscopy. This too can pick up fluorescence. You're also going to need a molecular probe. And these molecular probes can be attached to chemicals or to antibodies. And they can also be attached to molecules, molecules attached to quantum dots or nanoparticles. And both of these technologies also fluoresce. So here's an endoscopic image obtained using white light from a patient with Barrett's esophagus who had an area of high-grade dysplasia, biopsy-proven high-grade dysplasia. You'll note it's a very subtle architectural change here. There's a little bit of a minor impression in the dysplastic area. So here's the same image that was obtained using a scope in the fluorescence mode after the epithelium was sprayed with a fluorescent tagged peptide. Biopsies here from this area demonstrated high-grade dysplasia. When they then evaluated the biopsy that was now in a dish using a standard confocal microscope, not an endo microscope, you could see here that the fluorescently tagged peptide was binding to the Barrett's epithelial cells. A second method of molecular imaging that was published now just last year is now using fluorescent tagged lectins. And lectins were identified on the surface of the Barrett cells, and these were somewhat specific. And here's an endoscopic image obtained using white light in a patient with Barrett's esophagus who had an area of biopsy-proven high-grade dysplasia. You will note that on this image, there's no macroscopic abnormalities. I don't see any ulcers, nodules, mucosal defects. Can anybody see anything, Stu? No. No. Okay, so here's the same image where they tagged this fluorescent lectin. So in a normal area, you can see it's non-dysplastic. There's lots of green intensity. In the dysplastic area with less binding, it's showing up purple. When biopsies were taken from the purple area, it demonstrated high-grade dysplasia. Another molecular probe that's currently, it's more new kid on the block and in development, is actually using these quantum dots. And here's non-dysplastic Barrett's epithelial cells that have been immunostained with the DOS antibody, and the DOS antibody seems to be specific for Barrett's intestinal metaplasia. On the left-hand side is the DOS antibody that's now been conjugated to these quantum dots, and this was done in vitro, and then it was imaged with a fluorescent scope, and you can see that the quantum dots also have the ability to be used as an in vivo molecular imaging technology. So in summary, the genetic abnormalities that reflect early events in neoplastic progression seem to be much better than our standard use of dysplasia. Molecular biomarkers eventually may be better predictors of neoplastic progression than the dysplasia. And in vivo molecular imaging is early in development, but it does seem to hold promise to be an even better predictor of neoplastic progression than biomarker use alone. But please keep in mind that for now, that no biomarker has yet been validated in prospective clinical studies, so we still rely on the histologic finding of dysplasia for any clinical decision making in Barrett's esophagus. Thank you.